Welcome to the Ichthyology Research Laboratory in the Pontificia Universidad Javeriana. So, why do we study fish? Why did we choose fish to be our study object? Let's take a look at the percentage of participation in vertebrate groups. Here we can see the fish represent about the 50% of all of the vertebrate groups, giving it a high importance. There's also the ecosystems occupied by the fish. There are marine ecosystems and continental ecosystems. Here, in the Ichthyology Research Laboratory of the Pontificia Universidad Javeriana, we are mainly focused on continental ecosystems. So, why do we study freshwater fish as a study model? Let's try to answer. Why do we care about neotropical freshwater fish? First of all, we need to know that fish are physiologically restricted to the water bodies they inhabit, although they are isolated by terrestrial barriers. So, we have fauna taxonomically distinct. We also have phenomenal diversity of fish fauna. Studying freshwater fish, we get a better understanding of the evolution of all of the vertebrates. Also, we improve our understanding of historical changes in geological drainage patterns. Let's take a look at the percentage of representativeness of freshwater fish on Earth. On the left, we have the coverage of the planet Earth, being 29% of land and 70% of salt water. And here we see that just 1% is about freshwater ecosystems. In the right, we have in green all the saltwater fish species. They are about 58%. While in the left, we have in orange the freshwater fish species, about 41% only. So here we see that in really short amount of geographical space, about 1% of freshwater, there are about 41 freshwater fish species. So, having lots of species and individuals in really low space gives us a high diversity of freshwater fish. Here we can see some examples: the agujeto, Ptenoloceus beani, the anguilla, Cymbrancus marmoratus, the cascarudo, Oplosternum punctatum, the cucha, Ancistrus pijao, the bagrecito, Tatia sp, the pez cuchillo or knife fish, Apteronotus rostratus, among others. There's another highly important thing about fish, and is the involvement they have in the nutrient cycle in any freshwater ecosystem. They help in the transfer of nutrients, and they're also the regulator of different groups of animals and plants. So we see fish really involved in the food chain of ecosystems in freshwater. They are really involved with zooplankton and phytoplankton. We also see that many populations in Colombia use fish as their only source of protein. We also see the use of world's fish production. 71% is used for direct human consumption, while the rest, 29%, is used to manufacture different products intended for human consumption, especially flour and oil. So here we can see the high importance of fish production in the world. Now, in Colombia, there's another interest for fish, and it's the ornamental market. Here we can see that in the past year, 21 million of individuals were used in ornamental fish. So, what about us? We seek students willing to work in teams and interested in deepening the study of fish at the theoretical practical level, also to develop scientific and ichthyological skills. Here we have different academic levels of formation. With Dr. Saúl Prada Pedreros, we have master's student and undergraduate students. Also, with Dr. Javier Maldonado Campo, we have different academic levels, like doctorate, master's, and undergraduate degrees. We have 5 different research lines. The first, biogeography and population genetics. Second, functional and traditional ecology. Third, traditional biology. Fourth, taxonomy, phylogeny, and systematics. And fifth, scientific collections, curatorship, and restoration. Our objective as a research laboratory is to provide a space to initiate, expand, deepen, and improve the knowledge about neotropical freshwater fish to propose management and conservation plans. Let's take a look at the percentage of taxonomic representation at the Pontificia Universidad Javeriana Museum. First, we have the order Carassiformes with 63%. Then, we have the Siluriformes order with 19%. In third place, we found the Cichliformes with 13%. Fourth, we have Hypnotiformes with 3%. And in last but not least, Cyprinodontiformes with 2%. We can also see the percentage of samples collected by region. In first place, we have the Orinoco region with 77%. In second place, we found the Magdalena Cauca region with 12%. In third, we have the Amazonas with 8%. And in last, the Áreas Insulares and the Pacifico region with 3%. So, how we do it? First, we go to the field and collect the individuals. We follow different collection methodologies like atarraya, electrofishing, different type of nets, and snorkeling. After we have collected oral specimens and samples, we take them to our laboratory and to our collection in the museum. We also follow the Darwin Core archive components. This way we can have information organized and standardized. There are 2 mainly type of landscapes that we as a research laboratory go in the field. First, we have freshwater rivers, and in second, we have continental landscapes. As mentioned before, all the results of our investigations in taxonomy, systematics, biology, ecology, and biogeography are aimed in producing management and conservation plans. So, here we can see some of our results as a research laboratory. We have comparative phylogeography of freshwater fish in northwestern South America and lower Central America. We have the identification of areas of endemism in the Orinoquia of Colombia. We have mainland fish communities in the Amazon basin, a macroecological and genetic perspective. We also have comparative anatomy of bones, muscles, electric organs, and their discharge in Transcendian species of the family Sternopygidae. We have taxonomic revision of Eichmannia virescens and Sternopithecus macrurus from the Transcendian region in northwestern South America and the low Central America. We have molecular phylogeny of the genus Briconamericus inferred from nuclear and mitochondrial gene sequences. Functional diversity of assemblages of fish subjected to anthropic pressure in the middle basin of the Cusiana River. Areas of high endemism of fish in Colombian Orinoquia. Mathematical modeling of the operation of the reservoir of the Sogamoso River hydroelectric project. Taxonomic review of Grundulus bogotensis by means of the barcode gene, osteological descriptions, and geometric morphometrics. We also have chemical and metabolic investigations like the effect of an organochlorine pesticide on the mobility and production of acetylcholinesterase in Grundulus bogotensis, endangered and endemic species of the Cundiboyacense highland. We have the response of biomarkers in Grundulus bogotensis to the presence of heavy metals in the Bogotá River Basin and Suarez River Basin, Colombia. We also have the evaluation of the effect of anthropic activities on the biotic integrity of tributaries of the middle basin of the Cusiana River. And we have the regional altitudinal diversity of freshwater fishes north of the Andes, Colombia, among other investigations. Where can you find us? We are in the Ichthyology Laboratory 108B in the Building 53. Our two heads are Saúl Prada Pedreros and Javier Maldonado Campo. We also have alliances with other laboratories like the Population Genetics Lab, the Herpethology Lab, and the Limnology Lab, among others. Thank you. We wait for you.